Hello everyone and welcome. I'm the host of today's meeting, Jessica Tu from GenScript ProBio Marketing Department. On behalf of GenScript ProBio, I'd like to thank you all for attending today's webinar. GenScript ProBio is the subsidiary of GenScript Biotech Corporation, proactively providing end-to-end CDMO service from the drug discovery to commercialization with proactive strategies, professional solutions, and efficient processes in cell and gene therapy, vaccine, biologics discovery, and antibody protein drug to accelerate drug development for customers. This meeting will include two talks. One talk will take 30-minute presentation and 15-minute Q&A session. You can submit your questions at any point during the webinar using the chat or Q&A panel situated in the menu on the right. Now, I'm delighted to introduce today's first speaker, Dr. Li Chen. Dr. Chen is the Director of Biologics Discovery Department of GenScript ProBio. After obtaining his doctorate in immunology from the University of Massachusetts School of Medicine, he joined GenScript in 2015 and has been responsible for antibody drug discovery platform for the past 7 years. He has more than 200 project experience in antibody drug discovery and engineering innovation optimization, as well as 3 patents related to biopharmaceutical discovery. During today's webinar, Dr. Chen will be discussing accelerated ADC development via integrated antibody discovery platform. Now, I'm delighted to hand over to today's speaker, Dr. Chen.
Thank you, host. Just give me a second. Okay. Thank you everyone for attending this session. In this talk, we will discuss the strategies and the solutions for the discovery ADC candidates. During this presentation, I will first briefly go over the concepts and the technology trendings of antibody drug conjugates. Then we will have a detailed discussion on 2 crucial aspects of ADC discovery: how to find the suitable naked antibody leads, and how to evaluate the ADC candidates from the perspective of pharmacology effectiveness. Antibody drug conjugate is one of the fast-growing modalities in recent years. It consists of 3 parts. The antibody is a highly selective carrier for tumor-associated antigens that has differentiated expression level on cancer cells versus normal tissues, while the linker works as a switch to release the killing agent in designated environments. The payload is a chemical compound with high systemic toxicity designed to induce target cell death after being internalized into the tumor cell and released. When you put these 3 pieces together, it can guide the toxin to tumor tissues and kill the cancer cells. Comparing to traditional chemotherapies, ADC demonstrated unique features to avoid systemic exposure to the toxins, leading to less adverse effects. Currently, there are 15 ADC products that have been approved globally. It is half and half for solid tumor and blood tumor. In addition, there are more than 200 pipelines in clinical trials and another 200 plus pipelines in preclinical stage. United States and China are the biggest cancer therapeutic markets and takes about 80% of the global pipelines. So far, there has been three generations of ADCs. The first generation is comprised of traditional chemotherapy drugs and non-cleavable linkers. The major issues of these ADCs include undesirable efficacy from the toxin, unstable linker leading to systemic toxicity and a narrow dosage window, uh, hydrophobic toxin leading to negative impacts on the uptake of the drug. To overcome these weaknesses, people improved the toxin payloads in the second generation of ADC, but still relies on random conjugation, which makes it challenging to achieve homogeneity in terms of the number of payload molecules on each molecule of antibody. Nowadays, in the third generation of ADC, site-directed conjugation is widely used to overcome the DAR homogeneity challenge, and the concept of ADC has expanded from traditional chemical toxin to various uh, modules such as peptide, oligonucleotide, radioisotope, and even protein. In order for the ADC to kill the cancer cell, it needs to be transported into the target cell in which the toxin will be released. This process is called endocytosis or internalization. This unique feature brings several challenges in ADC discovery, including tissue penetration, target specificity, internalization efficiencies, timing of a payload release, the killing efficacy of a payload, etc., etc. As a result, a panel of factors needs to be considered during the discovery stage, such as choosing a suitable target with differentiated expression level on the cancer cell versus normal tissue, high affinity antibodies with desirable internalization capability, suitable linker and payload combinations, and also early stage pharmacology assessments for efficacy and safety in cells and in animal models. Next, we will move on to the first part of ADC discovery strategies, how to identify desirable naked antibody leads for ADC applications. So this is a general workflow of ADC discovery. Once you decide on the targets, a variety of technologies such as hybridomas, single B cells, phage libraries can be used to generate antibody leads. In the meantime, selection of a linker and a payload will also be made based on the indications, disease biology, and the MOA. Considering the potential safety risk, lots of people tend to choose toxins with plenty of clinical track record instead of those really novel ones. After obtaining antibody leads with desirable internalization features, a panel of ADCs uh, will be prepared in small scale, followed by cell-based assays and animal studies. At this point, the candidates will be narrowed down to less than half a dozen. These top candidates will go through antibody engineering, including humanization, affinity tuning, and FC engineering to gain particular features with optimized profile. Finally, in vitro and in vivo pharmacology model will be utilized again to validate the efficacy and the safety of the engineered final sequence, followed by developability studies to assess the product stabilities and homogeneity. The functional evaluation of ADC consists of two major aspects. One is the antibody internalization assay from the antibody Fab region, and the cytotoxicity from the payload toxin side. During the naked antibody discovery stage, we will first focus on the internalization parts mediated by antigen binding to the Fab region. The first option is live cell imaging-based internalizations. So basically, it utilizes a hardware called IncuSight to visualize the whole endocytosis process through fluorescent signals. The key features of this equipment is to generate internalization kinetics with a time course. As you can see from the case study of anti-ROR1 ADC in the bottom panel, for each defined concentration, for each defined antibody concentration of the benchmark UC961, there will be a curve reflecting the intensity of the internalization on the time axis. And you can include multiple curves to reflect the multiple curves in one graph to visualize the kinetics and the dose-dependent response. In addition, you can also generate a dose-dependent curves with fixed incubation time to calculate the internalization efficacy, the EC50s. Moreover, this assay can actually run with antibody supernatant, providing a high throughput approach to evaluate internalization capabilities during the early antibody screening stage. So by doing this, you can actually avoid losing potential candidates by making selections not solely based on the binding affinities. The second option is toxin conjugated antibody-based cytotoxicity assays. The DT3C is a synthetic toxin acting as a surrogate of a payload toxin in ADCs. Unlike typical toxin molecules, DT3C can associate with the Fc region of antibody through covalent interactions. After association with antibody, the whole complex will be uptaken by target cell, leading to the release DT3C toxin, which results in the cell killing. Since the binding between DT3C and the antibody is non-covalent, the chemical conjugation process, which is required for payload toxin in ADC, is no longer needed here. So basically, this assay is using the DT3C mediated cytotoxicity as a measurement of the internalization. Similar to the first option, this approach can also run in a high throughput fashion using antibody supernatant, providing a way to differentiate antibody leads in early screening stage. To demonstrate the robustness of this assay, we tested the DT3C system with the presence of a Herceptin and a negative control IgG on the right panel. As you can see from the graph here, SKBR3 cells gives a perfect dose curve of inhibition after incubation with Herceptin plus DT3C for 48 hours, while the negative control IgG, as illustrated as the black one, basically flat. The third option is pH indicator. The detection mechanism is also based on the co-internalization of fluorescent signal along with the antibodies, kind of similar to the first option, the lymphocytes. The pH rotor consists of an anti-Fc Fab fragment, this one, and a pH-sensitive dye which become highly fluorescent only in acidic environments, such as those of cell lysosomes and endosomes. The case study we did with the Herceptin shows decent dose response curve of fluorescent signals after incubation for 16 hours. The fourth option is cell surface fluorescence quenching. So it is a pretty straightforward flow cytometry-based assay to compare the relative amount of antibody entering the target cells. To put it in a simple way, the antibody will be incubated with target cells in two conditions in parallel, 4 degrees and 37 degrees. And the amount of the cell surface binding antibodies will be measured by typical fluorescent secondary antibodies. The signal difference reflects the relative amount of antibodies entering the target cells. The rationale of this approach is first assuming there is minimal internalization under 4 degree, and it also intentionally neglects other factors contributing to the fluorescent signal change, such as dissociation of antibodies from the target cell. As a result, this approach is not as accurate as those previous ones, but it requires the minimal hardware and reagent support, which makes it still a very popular option for early stage screening. So with all of these four options on hand, people will naturally wonder which one should I trust, right? So to answer these questions, we did a side-by-side comparison of these four assays using the from three ADCs. Two of them are already launched, while the third one is still in phase 2. Judging by the results, all four approaches gave similar ranking pattern of these three antibodies, and also similar range of EC50 values, except for the last one, the cell surface fluorescence quenching. This data demonstrated the reliability and the consistency between different assays. With that being said, you may still wonder which one should I choose. So we'll put together all of them in one table. The live cell imaging approach gives the best data and high throughput capabilities, which makes it the best choice for both larger scale screening and candidates characterizations. The DT3C toxin approach is also capable of high throughput and relatively low cost, making it an alternative option. The pH indicator is relatively expensive, while the fluorescence quenching method is not as good as the other in terms of the reproducibility. Next, let's move on to the second part of ADC discoveries, the pharmacology assessment of the drug antibody drug conjugates. So once you made the selection of the naked antibody leads, the next step is the payload and the link conjugations. Currently, there are a variety of payload and link used in different ADC pipelines. Here at GenScript ProBios, we can offer a library of chemicals, including more than 100 toxins and 700 plus linkers. as a reagent for proof of concept validation during the ADC discovery. In addition, you can also choose from traditional cysteine lysine-based conjugation chemistry or uh, site-specific conjugation approach to precisely control the DAR and the homogeneity. After the conjugation, ADC candidates will be first tested in vitro for cell-killing efficacies. This can be achieved through viability assays, apoptosis assays, or cell cycle assays. The case study done with HER2 showed on the right panel demonstrate that both ADC test articles, one of them is the reagent version of this 8201, both of them gives dose-dependent inhibition on the SKBR3 cells as measured by the cell titer glow reagents. In addition to direct inhibition, bystander effect is another MOA for ADCs. Bystander cells is the surrounding cells of the tumors, which could be antigen positive or antigen negative. When the tumor cells is lysed upon ADC function, toxin will be released and kill the neighboring cells. This MOA can be reconstituted in vitro as illustrated in the right panel. In the positive and the negative control group, testing ADC will be incubated with either antigen positive or negative cells, in this case, the HER2. And in the testing group at the bottom, conditional media from the drug-treated HER2 cells is transferred to the HER2 minus cell for efficacy evaluation. After all of these in vitro functional screening and validations, it is absolutely necessary to verify the efficacy and the safety of the ADC candidates in animal models. Typically, it includes three packages. For the efficacy part, people usually start with cancer cell line screening based on the indication and target expression profile, followed by tumorigenesis and efficacy tests. PK assay needs to be carried out both in vivo and ex vivo to determine the ADC stability, especially the linker parts and the controlled payload release. Toxicity studies is also a crucial part, including myelotoxicity assessment, tissue cross-reactivities, and payload-derived pathology. Here at GenScript Bio, we have established 24 tumor types with tumorigenesis from more than 70 cancer cell lines. In addition, we also provide 200 plus cell bank and a IHC panel for pre-study screening of a cancer cell line. PK and toxicity are crucial factors for the determination of ADC therapeutic windows, which is the constant import during ADC drug development. In the discovery stage, we usually pay attention to three checkpoints: the stabilities, target delivery, and the systemic toxicities. Stability can be assessed by ex vivo plasma stability tests and in vivo PK studies. Myelotoxicity assay using CD34 positive stem cell ex vivo. So at the end, let's come back to this process of ADC discoveries. There are a few key checkpoints that needs to be included. First of all, a high throughput internalization screening during the antibody leads generation stage will greatly help to weed out those non-desirable candidates. Secondly, pilot efficacy studies in animal models during early discovery stage gives us the opportunity to test the different combinations of naked antibody leads and payload linker in the actual disease model. Later on, after finalizing the candidate sequence, comprehensive PK/PD and the tox study, and also the developability assessments will greatly reduce the risk of failure in subsequent preclinical studies. So I think that will be all. Thank you everyone for listening. Let's move on to the Q&A session.
Thanks for Dr. Chen's excellent presentation. If you have any questions, please type it on the chat or Q&A panel. As you enter questions, please also follow our LinkedIn account, GenScript ProBio. There will be more online sharing resources, and we will share them on our LinkedIn at the first time. Thank you. Be translated into Vivo PK. Could you please answer this question?
Host, I think I lost your connection just for a few seconds. Can you repeat that question again?
Yeah. How ex vivo plasma stability can be translated into in vivo PK?
Well, I wouldn't call that like translation. And need to do some experiments. The stability test is one way to look at the linker stabilities, but that doesn't mean you can just skip the in vivo animal PK studies. We need both sets of data to make a comprehensive decisions about the candidates regarding the stabilities.
Yeah, okay. We also received some questions from the audience before, and let's take a look at the question. And the first question is, what is your recommended approach for assessing antibody internalization?
Well, as I mentioned in the second section of the talk, there are 4 different assays, which all of them are available at GenScript ProBio. But based on our experience with a few dozens of projects in last year, I think the Intel sites will be the top choice, while the DT3C is also a pretty good way to do the internalization in a high throughput fashions. But I do understand there are a lot of people still prefers the old-fashioned ways, the pH rotos. So most likely these 3 types of assays will be the recommended approach. But please note that the pH roto is pretty expensive, so usually people do not use it until the characterization stage.
Okay, thank you for your answer. And the next question is, do you have any site-specific compound conjugation technology for the ADC?
Well, for the discovery R&D stage, we do have some site-specific technology, site-specific conjugation technologies. Actually, you can buy those ready-to-use kit from off the shelf, like the one from Thermo Fisher's, and I believe there are a few other options out there. But all of these this site-specific conjugation technologies is actually IP protected. So if you're thinking about taking it to the commercial stage, the CMC clinical trials, I mean, you may still need to strike a deal with the IP owners for the commercial license.
Yeah, okay, sure. And the next question is how to quickly evaluate the developability of candidate antibody molecules?
Well, that's a really complicated question. So usually, I mean, for the ADC context, you need to look at from several different angles, including the internalization with naked antibodies, the killing efficacies after conjugation with the toxins, and also the chemical and physical stabilities of the ADCs after finalize the sequences, and also the safety issues. It's a constant pain point for most of the drug developer ADCs. Unlike traditional monoclonal or bispecific antibodies, the safety issues is always something that worries, concerns us during the preclinical development stage.
Yes, okay. And there is also a new question in the chat panel. It's compared to toxin-induced cytotoxicity of ADC. ADCC or CDC should be kept or more or less important?
Well, these are two distinct MOAs. Normally, we wouldn't put them together because if you imagine when you actually put dosing the ADC on the patient, the therapeutic windows is pretty low. I mean, the dose is pretty low for ADC, and the ADC dose is not even comparable to the dose when you treat a patient with a ADCC-driven drugs. So, put it in a simple way, if you are expecting a antibody to function through the ADCC, you need to a really high dose, which is impossible, almost impossible for ADC drugs. So usually these two MOAs are mutually exclusive based on the current therapeutic window of ADC. But you never know. Maybe in the future, there's people may find a way to compromise and combine these two MOAs together.
Yeah, okay. Thanks for Dr. Chen's answer.